Hey everybody! Welcome back to General Biochemistry Lecture. We're on Chapter 17, which is fatty acid catabolism. We're going backwards, so we did Chapter 19, and now we're going back to Chapter 17 and then Chapter 18. So you're not losing your mind. We are going backwards in the textbook. So, long-chain fatty acids are full of energy. They are more reduced than glucose. So when they are processed, they release a lot more energy. We have to first process them and make them into acetyl-CoA, which can then go into the citric acid cycle, yada yada. So oxidation of long-chain fatty acids actually serves as the central energy-yielding pathway in many organisms and tissues. So it can provide as much as 80% of the energy that you need for your heart and your liver. Electrons move from the fatty acids during oxidation, and they move to the respiratory chain. And of course, as we know, that drives ATP synthesis by ATP synthase. The acetyl-CoA that's made from fatty acids can be completely oxidized in the citric acid cycle. So, lots of fuel to be had. Beta oxidation is the oxidation of fatty acyl groups at the C3, carbon 3, or the beta position, hence the name, and that occurs after the carboxyl group at C1 is activated by attaching coenzyme A. So we're going to talk a lot about beta oxidation and what the different steps are, how we cycle through it. We're also going to talk about how we get fatty acids to the mitochondria where all of this is happening. Here are the key principles for this chapter. As always, go through them, make sure that you understand them, you can expound upon them, that sort of thing. We're going to start with the digestion, mobilization, and transport of fats. So there are several sources that you can actually get fatty acid fuel from. For us, we eat most of ours, so you can consume fat in the diet. We also store fat as lipid droplets in the cell, and those can be mobilized to provide fuel. Fats can be synthesized and then transported somewhere else in the body. And then there's autophagy. So if you have programmed cell death that occurs, you can kind of scavenge for parts, and one of the parts is the fats that are in the cell membrane and within the cell. You also scavenge for amino acids and other things of that nature to recycle. So this figure gives you a summary of the things we're going to talk about in terms of how fats are absorbed and how they get from, your mouth all the way to the cells that need it for energy or storage. So you eat something that has fat in it, and the first thing that's going to happen is bile salts are going to help emulsify those fats. Because remember, water plus fat, not going to do anything. They are not going to be digested well if you just put them in a bunch of acid. So the bile salts from the gallbladder help to emulsify dietary fats, and you form what's called mixed micelles, which just means fats, of different identities form a micelle. Intestinal lipases degrade the triacylglycerols, and those can get further broken down and taken up by the intestinal mucosa and converted into triacylglycerols again. At this point in time, we're going to see a protein called ApoC2. Apo just means that it doesn't have any ligand bound, and in this case, it's a lipoprotein, so it doesn't have any lipids bound. We're going to form what's called a chylomicron. We're going to talk about ApoC2 and chylomicrons in more detail, but this is just the overview. You incorporate triacylglycerols, cholesterol, and other apolipoproteins in this big blob called the chylomicron. And those can move through the lymphatic system and in the bloodstream to get to all the tissues and things that need fats. Once it reaches the capillaries, which are situated right next to the tissues that need the energy or are going to store the fats, it activates lipoprotein lipase, which converts the triacylglycerols to fatty acids and monoacylglycerols, or monoglycerides. The fatty acids can enter the cell, and then they can either be used as fuel or they can be reesterified, so made back into triacylglycerols and stored. So let's break down a few of the things that we introduced here. First, we're going to talk about the chylomicron. So what is it composed of? It's made of apolipoproteins, which means that they're proteins that don't have any lipid bound to them. And they target the triacylglycerols and all the things that need to be transported from one organ to another. So it helps to solubilize them and protect them and get them from one place to another. Chylomicrons have these apolipoproteins as well as cholesterol and all those triacylglycerols. Lipoprotein particles are just spherical aggregates of the apolipoproteins and the lipids. So it's arranged to where the hydrophobic lipids are at the core, just like when you think about a protein folding, you have hydrophobic residues in the center because they don't play well with water. So it is energetically favorable to hide them from water. And then you have things that are hydrophilic on the outside. So the hydrophobic lipids are going to be at the core. And then the hydrophilic protein surface is going to be on the outside protecting them, along with lipid head groups. So those are charged, right? So they have carboxylate tails. There's different densities of these particles, and I'm sure that you've heard of like HDL and LDL. Oh, LDL, that's bad, and HDL is good. The truth is, you actually need both. And so there's also very low density lipoproteins. So that's what the LDL is, low density, very low density, all the way up to very high density lipoproteins. And all that really tells you is how readily this little particle will give up all of the fats that it's holding. And when you think about transport, you want something that can keep the fat, protected while it's traveling through the bloodstream or wherever. But you also want it to be able to easily give up its contents when it gets to wherever it needs to go. So VLDL and LDL are not bad in and of themselves, but if you have more of that than you do HDL, it's more about your ratio, more than a certain amount of LDL. So that's one of the things that you'll probably learn a lot more about in medical school if you choose to go to medical school. But remember, it's the ratio. You need both. So here are a couple of proteins that are components of the chylomicrons. The primary protein component is apolipoprotein B48, but there's also ApoC2. And this protein is picked up in the blood by the chylomicrons from high-density lipoprotein particles. Once the chylomicron reaches the capillaries, which is right next door to the tissues that it needs to get into, it's going to activate lipoprotein lipase. This is an extracellular enzyme that's in the capillaries of muscle and adipose tissue. So we're talking about muscle tissue that's going to burn the fat, and adipose tissue is going to store it. And it hydrolyzes the triacylglycerols to form free fatty acids and monoacylglycerols, which again, I may also say monoglycerides. Same thing. Free fatty acids, you may see abbreviated as FFA, particularly in your textbook if you choose to read the chapter. So, lipoprotein lipase hydrolyzes triacylglycerols to free fatty acids and monoglycerides and is activated by ApoC2. And then after that, you have entry of the fatty acids into the cell, and then from there, the cell determines what it needs to do. So fatty acids are converted to triacylglycerols in the liver, and this is for excess fatty acids. So your body has used all that it needs, and now the liver is going to do the work to take care of the rest that you have consumed or the rest that you have in your body. Those triacylglycerols are packed with apolipoproteins into VLDL, so that's the very low density ones, and they're secreted and transported in the blood to adipose tissue. They're removed, stored in lipid droplets, bam. Now you could see where there could be a problem, right? Let's say that you consistently have extra fat, whether it's because you eat an extremely fatty diet and you can't, use all of it, or because your body makes a lot of fat and transport a lot of it, what have you. If you have a lot of those VLDLs floating around because you've got excess fat and you're trying to send it to the adipose tissue, then maybe some of that gets deposited on arteries and things like that, which is bad. So that's why looking at how much LDL you have in relation to HDL is kind of a marker for, okay, maybe this could be a problem. Just like everything else, hormones trigger mobilization of those stored fats. The lipid droplets that are in your organelles, they can be mobilized. So they have a core of triacylglycerols and sterol esters. Sterols, think about cholesterol and hormones. So things that synthesize steroids. All of those things are surrounded by a monolayer of phospholipids. Now, we didn't talk about phospholipids when we covered fatty acids, but phospholipids just have phosphate groups on their lipid head, so on that carboxylate. You don't need to worry about the structure of it. But the phospholipids are hydrophilic, and the phosphate head groups are going to be poking out into the cytosol, and then the lipid tails are pointing in, hanging out with all of the stored fat. There's a family of proteins that also coats the surface of lipid droplets, and those are perilipins. They restrict access to those lipid droplets, and that prevents mobilization before it's time. Once you have a hormonal trigger, which if you think about what state your body would be in when it says, hey, we need some energy, right? It means that you're doing some kind of workout or fight or flight, something like that. So we're going to have glucagon and epinephrine signaling for the mobilization of stored fats. Glucagon, remember, acts in the liver. Epinephrine acts on muscle. Once these hormones are released and they meet their receptor on the cells, then you trigger adenylyl cyclase through the heterotrimeric G protein activation, and you create cyclic AMP. This activates pKa. This should sound very familiar because we talked about these first couple of steps when we talked about mobilization of glycogen to make glucose. So that same signal will also trigger mobilization of stored fat. Once pKa is triggered, it goes and phosphorylates things, and it leads to a signal cascade that will mobilize fat. And that fat gets released into the bloodstream so that it can then reach all the tissues that need it. So the same way that the chylomicrons and things will kind of pick up circulating fatty acids and then make sure it gets to its proper location, that's what's going to happen here too. Once you get those fats to a cell that needs them, you're going to start oxidizing them to make acetyl-CoA. One of the things that helps too with making sure that fatty acids reach their destination is human serum albumin. And it will bind non-covalently to the free fatty acids that are in the blood. So you have a lot of this in your blood, a lot of this protein. And that will, again, get those free fatty acids to their destination. And it helps to protect them because there are different areas where the fats can bind and kind of be tucked away from the blood. So what happens with all of those fats? Most of the energy that's available in a triacylglycerol is in the long chain fatty acid. There is that glycerol that can be converted to glyceraldehyde 3-phosphate, which should sound familiar. That is an intermediate in glycolysis. So we know that glycolysis does not make a whole lot of ATP in comparison to the other processes that we talked about in chapter 16. But there is a pathway to take glycerol, phosphorylate it, and then use the same glycerol 3-phosphate dehydrogenase, which is in the cytosol, to create dihydroxyacetone phosphate. And then we use that isomerase to create glyceraldehyde 3-phosphate. Once that happens, you can continue on and make some ATP. So all of the fatty acids need to get transported into the mitochondria where beta oxidation occurs. If that fatty acid has less than 12 carbons, it's just going to freely diffuse across the membranes. However, a lot of the fatty acids that we consume that need to be oxidized are longer than that. So for these fatty acids, you have to use the carnitine shuttle. And that will transport the fats through the mitochondrial membrane. Before that happens, we have to activate the fatty acyl-CoA and attach it to a carnitine. The enzymes that do this are fatty acyl-CoA synthetases. They use ATP, and they are present in the outer mitochondrial membrane to activate the fatty acid to convert it to a fatty acyl-CoA. So that's the first thing that needs to happen. There's lots of different isozymes because of the varying chain lengths for the fatty acids. This is a very high energy compound that we make, and it is a reaction that is favorable because we're conserving energy with that bond to the CoA. And it's made even more favorable because we're actually releasing pyrophosphate. So when we activate the fatty acid with ATP in the first step, we're actually breaking this bond here. Normally we use the gamma phosphate, the one that's on the very end. We do that hydrolysis by breaking this bond here. But we're not doing that this time. So we're going to release pyrophosphate. And that pyrophosphate immediately gets hydrolyzed to form two inorganic phosphates. And that is very favorable. In addition to that, we're going to swap out that AMP for CoA. So the thiolate can attack this carbon, and then the leaving group is AMP. So the overall reaction for fatty acyl-CoA formation, it involves 3 steps. There's a 2-step formation of the fatty acyl-CoA. You activate with ATP, and then you transesterify with CoA. The third part is the hydrolysis of the pyrophosphate. Now that we've got our fatty acyl-CoA, we can now attach it to carnitine, which is shown here. Carnitine is going to help with shuttling this fatty acid all the way into the matrix. Now, there's a couple of carnitine acyl transferases. I may also call it CPT1 because it's also called carnitine palmitoyl transferase 1, but it actually works on more than just palmitate. So excuse me if I call it CPT1. What this enzyme does is it takes that fatty acyl-CoA that we just made and it attaches it instead to the hydroxyl of that carnitine group. So you're swapping CoA for carnitine. Once that happens, we can now transport this fatty acid into the matrix. So to review what we've talked about so far, we've got free fatty acids that are in the cytosol that end up getting a CoA attached to them. That CoA is then swapped out for carnitine. And once that happens, then we can have transfer from the intermembrane space, which remember, the intermembrane space is just kind of like an extension of the cytosol. Things can kind of go back and forth freely, but things cannot freely cross the intermitochondrial membrane. So we've got this carnitine attached fatty acid, which can then go through a transporter that is specifically for carnitine and acylcarnitine. It's the acylcarnitine-carnitine cotransporter. It allows the acylcarnitine into the matrix, and it allows one carnitine to move out into the intermembrane space. So you're swapping one for the other, so you always have free carnitine available to do just that, to be available to that carnitine acyltransferase 1 to make the swap. Once that carnitine, the acylcarnitine is in the matrix, we have to do another swap. And that's swap with a carnitine acyltransferase 2. So it transfers that fatty acyl group back onto another CoA. You're generating that carnitine that is then going to go back out into the intermembrane space and pick up another fatty acyl group. This enzyme is located on the inner face of the intermitochondrial membrane. And that's important to note because we don't want to be exchanging that carnitine before the fatty acyl group makes it into the matrix. If you do, then you just did all that chemistry and all that work for nothing, because that fatty acyl group attached to a CoA cannot pass through the inner membrane. So there's 2 pools of coenzyme A in the cell. There's one pool that's in the cytosol, and there's another pool in the mitochondria. We keep these things separate because they have kind of 2 different roles. So the CoA in the mitochondrial matrix is by and large used for degrading things. So pyruvate, we've seen it. Fatty acids, we're seeing it now. And we'll see amino acids to come. The CoA in the cytosol is actually used in making fatty acids because coenzyme A is a great handle to hold on to. And it's something that can be, that enzymes can use and kind of hand off from one enzyme to the next. So it makes repeated rounds of adding on acetyl groups a lot easier. The carnitine shuttle is a huge point of control. It's the entry into the matrix, and once that fatty acyl-CoA gets into the matrix of the mitochondria, there's no return. It's going to get converted into acetyl-CoA through beta oxidation. So, the rate-limiting step for this process of converting fatty acids into acetyl-CoA is actually the carnitine-mediated entry. So it is rate limiting. If you don't have the fatty acyl groups in there in the matrix, you can't do the chemistry. So if you control how much is in the matrix at a given time, then you can control how much energy you're producing. CPT1 is also inhibited by malonyl-CoA, which is an intermediate in fatty acid synthesis. We don't want to have synthesis and breakdown happening at the same time because that's pretty wasteful. It's just like with glycolysis and gluconeogenesis. You don't want to have both of those things going on at the same time because you're consuming the very energy that you're making. Now that we've got the fatty acyl group into the mitochondrial matrix where all the enzymes live for beta oxidation, we could talk about oxidation of fatty acids. So the first stage is beta oxidation, which is what we're going to cover. Fatty acids undergo oxidative removal of 2 carbon units in the form of acetyl-CoA. And you just kind of keep chugging through until you've made all your CoA. Along the way, you're also making NADH and FADH2. And since we've already talked about the citric acid cycle and oxidative phosphorylation, we already know what happens to those molecules because they're universal electron donors. They end up giving up their electrons in the electron transport chain, which ultimately converts all of that energy from the electrons and the flow of electrons into ATP synthesis. So the second stage would be oxidizing all those acetyl-CoA groups to carbon dioxide in the citric acid cycle. And we're generating more NADH, more FADH2, and GTP, which can ultimately get converted to ATP. And finally, electron transfer, oxidative phosphorylation. So we already know the second stage and the third stage of fatty acid oxidation. We need to learn more about the first stage, which is beta oxidation. The first thing that needs to happen is we need to create a double bond. That is going to be done by acyl-CoA dehydrogenase. It's a flavoprotein that's got FAD really tightly bound, and it dehydrogenates the fatty acyl-CoA to make a trans-enoyl-CoA. So when you look at the highlighted region, that's where you're going to act. That's where the chemistry is going to happen. And we're actually going to make a double bond here. So this is the beta carbon, and that's why it's called beta oxidation. There's FADH2 that's created here, and it will ultimately give up its electrons to the respiratory chain through an enzyme that we're going to talk about in just a second. I also want to note that we're making a trans bond here. So that means that we've got groups on opposite sides of that double bond. There's different isozymes for the acyl-CoA dehydrogenase, just like there were for the enzyme that creates the CoA and the carnitine. Isozymes are specific for fatty acyl chain lengths. So we've got very long, medium, and short. And all of these enzymes are located in the matrix, which makes sense because that's where beta oxidation occurs, right? So as a part of creating this double bond, this trans-enoyl intermediate, we also generate FADH2, which we already know is going to ultimately drop off its electrons in the respiratory chain. But how does it get there? The ETF ubiquinone oxidoreductase. It accepts electrons from ETF, and then it passes those electrons to ubiquinone. I'll say here too, you should also think about once those electrons have reached ubiquinone, coenzyme Q, where do they go next? We already talked about the citric acid cycle and oxidative phosphorylation, so you should know where they go next. Okay, so now that we have, so also let's talk about comparison between citric acid cycle and beta oxidation. There's a lot of repetition here in terms of the chemistry, which suggests that there may have been some kind of gene duplication event and then evolution of specificity for different substrates over time that caused these two pathways to be quite similar. So when we have acyl-CoA dehydrogenase, we can also look at succinate dehydrogenase and look at those enzymes and see how similar they are. So both of them are in the inner membrane. There's a double bond that's introduced into a carboxylic acid between the alpha and beta carbons. Remember that the alpha carbon is carbon 1, beta carbon is carbon 2. And then we've got I guess I could erase this. So the carboxylic acid, the one that has the CO double bond that's also bonded to the CoA, that one's carbon 1. Carbon 2 and 3 is alpha and beta. We also use FAD as an electron acceptor in both of these enzymes. And ultimately, those electrons end up getting passed through the respiratory chain and they end up reaching molecular oxygen. So lots of similarities here in terms of location and the chemistry that they do and the fact that they can donate electrons. Okay, now that we have this double bond that's introduced, we can have hydration of this double bond. And that's catalyzed by enoyl-CoA hydratase. Again, I want to stress the fact that it is a trans intermediate that the enoyl-CoA hydratase works on. Once you do that, you form the beta hydroxyl-CoA, and it's in the L form. This reaction is analogous to the fumarase reaction in the citric acid cycle. This is the point where we are making some NADH when we take this hydroxyacyl-CoA and use a dehydrogenase to do the next step. That NADH, again, it can go off and donate its electrons in the in the electron transport chain, which will ultimately make ATP. And what this enzyme does is it catalyzes the dehydrogenation of the beta hydroxyl acyl-CoA to a beta keto acyl-CoA. This enzyme is very specific for the L stereoisomer. So that's why I was stressing trans, that the double bond is a trans double bond in that first step. Then we have enoyl hydratase come in and hydrate the bond, and we have the L isomer that is created. That L isomer is then dehydrogenated. This is very similar to malate dehydrogenase in the citric acid cycle. We know that the electrons from NADH can be shuttled into the electron transport chain, And where they enter is complex 1, the NADH dehydrogenase. It will accept all those electrons that are created by the reaction that we just covered, by the beta hydroxyacyl CoA dehydrogenase. The last step is we have to liberate acetyl-CoA, and that is done by thiolase. You'll also see it called acyl-CoA acetyltransferase. Thiolase is a lot easier to say. So that catalyzes the reaction of the beta-ketoacyl-CoA with free CoA, to yield acetyl-CoA, and then you have a fatty acyl-CoA that's shortened by 2 carbons. This reaction is the reverse of a Claisen condensation. So with the Claisen condensation, you're taking 2 esters and putting them together, or you're taking one ketone group or carboxylic acid, and you're taking an ester and you're smashing them together. So this is the reverse. You're breaking it apart. Just like with the citric acid cycle and with oxidative phosphorylation, we saw that some of the enzymes that do work on intermediates, from one another, hang out together. And we also saw this with the PDH complex that would make acetyl-CoA from pyruvate. What we have is the trifunctional protein. It's a multi-enzyme complex that's associated with the inner mitochondrial membrane, and it catalyzes steps 2 through 4 of beta oxidation for fatty acyl chains that are 12 or more carbons. So that means that you can channel your, you can substrate, let me try that again. You can channel your substrate from one enzyme to the next without losing your intermediates. And that's really important. This is a very big complex. It's a heterooctamer, so it's got alpha and beta subunits, and it's got 4 of each of those. The alpha subunits contain the enoyl hydratase and the hydroxyacyl-CoA dehydrogenase activity. The beta subunits catalyze that 4th reaction, the thiolase reaction, where you are cleaving with free CoA. So let's talk about the chemical logic of beta oxidation. The first 3 reactions convert a stable single bond between methylene groups to something that's much less stable because of the groups that we formed around it. The ketone function on that beta carbon, carbon 3, makes it really great for nucleophilic attack because it's an electrophile. It's partially positive. We also know that CoA makes a great leaving group. So we've got this less stable bond. We've got a great leaving group. That means that it's a lot easier to break the bond between the alpha and beta carbon. This is a look at the beta oxidation, citric acid cycle, and the oxidation of isoleucine, which also happens with leucine and valine. We haven't covered that yet. But it's just a look at the conserved reaction sequence. So you form a double bond using flavoprotein, and you generate FADH2, which can then go off and give its electrons up to the respiratory chain. You hydrate this double bond, and then you end up creating a ketone, creating NADH, which again gives up its electrons, and now you've got a carbon that is much easier to attack, and you can carry on doing the rest of your chemistry. So these steps in beta oxidation are repeated to yield acetyl-CoA and ATP. So you're going to shorten that fatty acyl-CoA by 2 units every time until all of that fatty acyl-CoA has been turned into acetyl-CoA. So if you had a 14 carbon fatty acid and there were no double bonds, which we're going to talk about that in a minute, if there's no double bonds, you can just keep going through beta oxidation, going, and you end up making 7 acetyl-CoA molecules. So each pass, just to reiterate, you're going to remove 1 molecule of acetyl-CoA and 2 pairs of electrons and 4 protons. 2 pairs of electrons means 4 electrons. And remember that we're doing that in the form of FADH2 and NADH. So that's every single round of oxidation. If we look at the entire reaction, and let's say we're using palmitoyl-CoA, then you're going to need all these cofactors. So you need a lot of CoA, FAD, NAD, and you also use water, right, for that hydrolysis reaction to make 8 acetyl-CoAs, because palmitoyl-CoA has 16 carbons. And you're making 7 FADH2s, 7 NADHs. You get the idea here. You don't need to memorize this overall equation, but you do need to know the products. You don't even really need to know the hydrogens, okay? I just want you to know acetyl-CoA, FADH2, and NADH. How much will you make for a given fatty acid going through beta oxidation? So each of the FADH2 molecules ends up generating about 1.5 molecules of ATP in terms of the energy that it provides to create ATP using ATP synthase. And NADH generates about 2.5 molecules of ATP. So in total, since we have 1 of each of these made through beta oxidation, you make 4 ATP for each pass through beta oxidation. So that's another question I could ask you. How much ATP is generated by the complete oxidation of a fatty acid of this number of carbons? So if we look at the real overall reaction, starting with beta oxidation, and then we include all the transferring of electrons and oxidative phosphorylation, then we actually end up making 28 ATPs from palmitoyl-CoA. So we just covered that acetyl-CoA can be completely oxidized through the citric acid cycle and oxidative phosphorylation. So when we do all of that calculations, because we've got all that ATP that we can generate from all the acetyl-CoA, we're actually making a lot of ATP. So notice the difference here. We're not including the acetyl-CoA in the overall, this is the overall complete reaction to go from palmitoyl-CoA to as much energy as we can possibly get, and we are creating a bunch of CO2 in the process. So you should be able to get from the fatty acid to how much ATP you make overall from the complete oxidation of that fatty acid. So this just gives you an idea of how many ATPs are yielded at each step based on, how many times you go through the steps. So FADH2, you make 7 of them if we're using palmitoyl-CoA as our example. And ultimately you end up forming 10.5 ATPs. So this just goes through and walks you through how many ATPs are formed by each step and how we get to that 108 ATPs total. So walk through that, make sure you understand how we get that total. So a lot of the fats that we eat are actually unsaturated, which means that we need to do some extra chemistry before they can enter beta oxidation. Or once you reach a double bond that is not in a favorable position, we've got to do some chemistry to make that different before we can move on. Enoyl-CoA hydratase can't catalyze the addition of water to a cis double bond. So if there's a cis double bond, like, I don't know, in naturally occurring fats, then we've got a problem. So we've got to do 2 additional steps. There's enoyl-CoA isomerase, which is going to convert cis double bonds to trans double bonds. And then that enoyl-CoA hydratase can work on it if that double bond is in a good position. Otherwise, we're going to also have to use a reductase that reduces cis double bonds. So let's talk about a monounsaturated fatty acid first. Let's say that we've got 1 double bond, it's at carbon 9. That means that we can go through a few rounds of beta oxidation first. Then we hit a snag. We've got this cis double bond here, and enoyl-CoA hydratase can't do anything with it. So instead, we're going to call our friend isomerase, which is going to take that cis double bond and make it trans. Now we can keep going through beta oxidation. La 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 la. But what happens if you have a polyunsaturated fatty acid, which means that we've got more than 1 double bond? That's when you're going to need the isomerase and the reductase. So let's say that we have an 18 carbon fatty acid, 2 double bonds. We can go la 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 la, do beta oxidation until we hit our first snag. We've got a cis double bond. We can't do anything with it. It needs to be trans. So we use the isomerase to create that trans double bond. Now we have a double bond between the alpha and beta carbons. And that's what we want anyway. So we can do another cycle of beta oxidation. And then we can even do another round of oxidation of a second cycle of beta oxidation. But then we hit another snag. We've got this double bond, which is okay, but we've got this double bond here, which is trans, and we can't do anything with this. So we have to use a reductase, and that's going to put a double bond between carbons 3 and 4. Then we use the isomerase again to create that trans double bond between carbons 2 and 3, the alpha and beta carbon. Now we can continue through beta oxidation with no problems. So here what I want you to know is when you look at a fatty acid, are you going to need the reductase and the isomerase, or just the isomerase? So you need to know when you need those enzymes and how many rounds of beta oxidation you can get through before you have to pull out the isomerase or the reductase. We've got another complication. Occasionally, you have odd number fatty acids. Not a super high occurrence in nature, but it does happen, and we have to do something about that because acetyl-CoA is a 2-carbon molecule. If we have an odd number of carbons in a fatty acid, we're going to end up with an odd number at the end. We're going to have an extra carbon. So we actually have to do an extra 3 reactions just to rectify this. So there are cattle and other ruminant animals, meaning that they kind of go and graze on the grass. They create propionate, which is a 3-carbon compound. If you remember your prefixes from organic, prop, that's 3 carbons. Odd number fatty acids are oxidized through beta oxidation, and they yield all the acetyl-CoA that they can make, and then propionyl-CoA. So we have to do something with that molecule. Propionyl-CoA carboxylase will carboxylate propionyl-CoA to make methylmalonyl-CoA. It uses biotin, which remember is our cofactor that will help us move 1 carbon units in the form of carbon dioxide onto a molecule. So I'm not going to require you to draw this reaction, but it is something that you should be familiar with because it's something that we have drawn in the past. Once we make that methylmalonyl-CoA, we can use an epimerase to go from the D form to the L form. And that's important because remember, there are enzymes in the beta oxidation cycle that require specific stereochemistry. So now we've got the L form of methylmalonyl-CoA. And note how many carbons it has. The next thing we can do is use a mutase that's going to catalyze the rearrangement of L-methylmalonyl-CoA to succinyl-CoA. So instead of there being this carbonyl here attached to this carbon, we're moving it all the way to the end. And this is done with coenzyme B12, which is also called 5'-deoxyadenylcobalamin. And it's a really cool coenzyme. Not going to require you to know that chemistry, but it uses radical chemistry, which is something that you don't get to see very often. If you end up taking a higher level biochemistry course, you will definitely encounter this mechanism and you will have to draw it. But I feel like it's a little bit above our pay grade since this is our first introduction to biochemistry. But if you're interested, there is a little insert in the textbook that talks about it and how you form a radical within the coenzyme itself, and you have this separation, this homolytic cleavage, and then you can donate an electron to whatever it is that you're doing and reform a carbon bond. So it's pretty cool. But all you need to know for this class is that you have methylmalonyl-CoA mutase. And remember, mutases are similar to isomerases. They're a subclass. They group transfer within the same molecule. If you have a deficiency in propionyl-CoA carboxylase, which is the one that adds on the 1 carbon unit using biotin, then you have what's called propionic acidemia. And this occurs in about 1 in 100,000 babies. So when you have a child, there's all these newborn screens that happen. So they draw blood, and it hurts your heart because you never want to hear a baby cry. And they're pricking your little baby's foot and drawing blood, and it's a whole ordeal. But they screen for these rare but very life-threatening diseases. And this is one of the things that's screened for. So you can look at, the genetics of this baby and see what alleles they have. You could also do testing for enzymes and all kinds of things. So that newborn screen that happens right after baby is born, about a day or so after they're born, it's a terrible thing to witness, but it's very, very important because though these things are rare, if your baby is afflicted, then there are dietary restrictions and changes and things that have to happen immediately in order to give baby a long, happy, healthy life. So in this case, this deficiency can cause terrible acidification of the blood and the urine because you have all this propionyl CoA from odd numbered fatty acids that you can't do anything with. And so it ends up being released into the blood as propionate, and it can get through using the carnitine shuttle. So propionate is just a very, very short fatty acyl CoA that you can, swap out carnitine just like any of the other fatty acids. So it's bad news. This wouldn't be a biochemistry lecture if we didn't talk about transcription factors. So we've already talked about a signaling pathway using hormones. We've talked about a metabolic pathway and all the chemistry. Now we've got to talk about transcription factors. It's going to be a theme that you're going to see for pretty much the rest of all these metabolic pathway lectures. So there's a whole family of nuclear receptors called the PPAR family. And these are transcription factors that affect a lot of metabolic processes. And they respond to different fatty acid-like ligands. So fatty acids are important for forming these. And these receptors will bind to fatty acid-like ligands. And the alpha subfamily will stimulate the synthesis of the enzymes required in beta oxidation when there's increase in demand for energy from fat catabolism. So when this PPAR alpha binds to its little spot on the genome, then it will stimulate synthesis. So that means you're increasing the messenger RNA transcripts that are made, and you are making more of the enzymes required for beta oxidation to meet demand. Let's talk about some other genetic defects, because with something that's so important like beta oxidation, mutations that cause changes in functionality or deletion of genes or anything like that can be embryonic lethal, or you can have a severe defect. So medium chain acyl-CoA dehydrogenase, that's one of the enzymes that is required for making fatty acids with that the CoA on the end. And if you have two mutant alleles, then you can't actually oxidize medium chain acyl-CoAs. So the symptoms include fatty liver, because remember, all of that fat gets stored as lipid droplets, and the liver is like, one of those storage places in addition to adipose tissue. You have high levels of octanoic acid, which is a medium chain acyl-CoA. You can actually be comatose, and you can die from this. So it's a very big ordeal. Beta oxidation is not the only, it occurs in more than just the mitochondria. It also occurs to a certain degree in peroxisomes, which we have peroxisomes, and plants also have peroxisomes. All of the steps are present, but there's a couple of differences. The first difference is that in the mitochondria, the first step of creating this enoyl intermediate, you're making FADH2, and then those electrons can be shuttled to the respiratory transport chain and ultimately end up making ATP. In the peroxisome, that FADH2 is donating its electrons directly to oxygen and forming hydrogen peroxide, hence the name peroxisome. That hydrogen peroxide can then be made into water and oxygen by catalase. It's an enzyme, which you may have done like a little experiment with in undergrad, or even in elementary or middle or high school, seeing something go fizzy and make oxygen. Another key difference is what happens with the dehydrogenase step here, where you're making NADH. That NADH in the mitochondria can go on to donate its electrons similarly to FADH2, whereas the NADH that gets made in the peroxisome has to be exported so that it can be reoxidized and then used again. Another difference is what happens to the acetyl-CoA at the end. In the mitochondria, well, the citric acid cycle is right there, so why not? However, with the peroxisome, you're not doing the citric acid cycle there. That's something that happens in the mitochondria. So you export the acetyl-CoA, and it can be used in the cytosol for fatty acid synthesis, or it can make its way into the mitochondria for citric acid cycle and ultimately give up its electrons in that route. So we already covered the catalase reaction, where we're taking the hydrogen peroxide that's created in peroxisomes and making water and oxygen. The peroxisomal system is much more active on the very long chain fatty acids and branched chain fatty acids. So while it's not as active overall as the mitochondria, it's still very important for breaking down fatty acids, because it specifically breaks down these really long fatty acid chains and the branched ones. We're going to talk about some more defects associated with beta oxidation, this time oxidation in the peroxisome. So Zellweger syndrome is characterized by an inability to make peroxisomes. So you don't have any of the metabolism that's related to peroxisomes, which is real bad news. Then there's XALD. I'm not even gonna, you know that I struggle sometimes with these medical names. I could never be a medical doctor because my tongue just revolts and says, oh no, we're gonna do this backflip over here. We're not gonna say that word. So long enzyme names, I'm usually okay with, but there's something about the genetic defects and different diseases and stuff that it just doesn't jive with me. The chemistry is much more my bag. So I'm just going to stay in my bag and let y'all do the other. Y'all go and be the doctors and PAs and know how to say all those words. So this defect is characterized by the inability of peroxisomes to actually oxidize those very long chain fatty acids. And that's because there's a transporter that isn't functioning properly. So just like with the mitochondria, you have to transport the fatty acids into the peroxisome so that they can be processed. If you can't get the substrate to the enzyme, then you can't have the reaction. So you can have all of the enzymes for beta oxidation, but if you don't have the substrate, you're not doing it. Now we've got to talk about ketone bodies, which you may have heard of. Ketone bodies actually refers to three different things: acetone, acetoacetate, and D-beta hydroxybutyrate, which I'm just going to shorten to hydroxybutyrate because saying the D-beta part for some reason is just a lot of extra. So I'm talking about D-beta hydroxybutyrate when I say hydroxybutyrate. Ketone bodies is kind of like a antiquated term. All of these don't even have ketones because hydroxybutyrate doesn't have a ketone at all, but it's related to acetoacetate. Ketone bodies are formed from acetyl-CoA in the liver. Only the liver does this. The acetone that's created is exhaled. The acetoacetate and the hydroxybutyrate are transported to tissues that are not the liver, which is what extrahepatic means, and converts, the other tissues will convert acetoacetate and hydroxybutyrate to acetyl-CoA, which can then be oxidized in the citric acid cycle. So this is a normal thing that occurs. The way that we get to ketone bodies is by reversing the reverse Claisen condensation. That's the last step of beta oxidation. So you're doing a Claisen where you're taking two acetyl-CoAs together, smashing them together. You have a CoA as a leaving group, and you make acetoacetyl-CoA. So it's a 4-carbon molecule attached to CoA. From there, HMG-CoA synthase will take acetoacetyl-CoA and add on another acetyl-CoA, and you make HMG-CoA. So hydroxy beta methylglutarol-CoA. That's a mouthful. HMG. Then there's a HMG lyase that cuts. So lyase, always think cut. And it catalyzes the cleavage of HMG-CoA to free acetoacetate and acetyl-CoA. This probably seems kind of ridiculous. Like, why would you add on another acetate or acetyl-CoA and then cut it? But the reason is you have options, right? So sometimes you want to make HMG-CoA. Sometimes you want to break it down. So you need to have those options. And acetoacetate is something that the cell can use to do different things. So acetoacetate decarboxylase, you could probably guess, removes carbon dioxide. So it will remove a carbon in the form of carbon dioxide, and then you get acetone as a product. That acetone we exhale out. The hydroxybutyrate dehydrogenase will catalyze the reversible reduction of acetoacetate to hydroxybutyrate. This can actually happen in all of the tissues outside of the liver. Then we also have beta ketoacyl-CoA transferase, which will activate the acetoacetate, just like we activate fatty acids with that acyl with the CoA. Same thing with acetoacetate. And once it is activated, you can cut it with the thiolase, and then you now have 2 acetyl-CoAs. So this can happen to use up all of that hydroxybutyrate as fuel in the citric acid cycle. The liver makes these ketone bodies, but it cannot actually consume them. It doesn't have the ketoacyl-CoA transferase. So that means that it can't activate the acetoacetate, which is required to actually use it for anything else. So it makes it, ships it out, and says, okay, I'll take the hit on this one. All of y'all tissues, take these ketone bodies. Be happy. Now, we don't make a lot of ketone bodies, okay? That if you have a normally functioning metabolism, you're not going to be making a bunch of ketone bodies. However, during starvation, or if you have diabetes that is uncontrolled, you will go into hyperdrive with making ketone bodies. So when you accumulate acetyl-CoA, then your body starts making ketones, these ketone bodies. And part of the reason is because you've got all this acetyl-CoA, you have to do something with it. You can't, your other tissues don't have the capacity to deal with a lot of acetyl-CoA. As a result of formation of ketone bodies, you can actually go into acidosis, which is caused by increased levels of acetoacetate and hydroxybutyrate in the blood, because you're making all of this and it's being exported by the liver, and then your cells are like, look, man, we already got enough. I don't know what you're going to do with this, but stay out of here. So you're going to lower the pH of your blood, which is bad. And you can also go into ketosis, which means that you have a lot of ketone bodies in the blood in your urine. And the two of these things together is called ketoacidosis, where you have both of these conditions at the same time. This is something that's common when you're looking at uncontrolled diabetes. So we have acetoacetate decarboxylase, which we covered, that decarboxylates acetoacetate to make acetone. We exhale out that acetone. Under normal circumstances, that's fine. But if you have a lot of this activity, then it could actually make your breath smell pretty bad. And it's a very characteristic odor that can be used to kind of help diagnose someone with untreated diabetes. That's it for chapter 17. We just covered all that you need to know about fatty acid oxidation. There are some other things that we did not talk about, particularly with the peroxisomes. If it wasn't in the lecture, don't you worry about it. But if you're interested, by all means, go forth and conquer. I hope that you enjoyed this one, talking about some of the diseases and making some links to the citric acid cycle and how they are kind of related. They do some very similar chemistry. So in terms of preparation, be thinking about how you can draw those, think about those characteristics that you can compare beta oxidation and the citric acid cycle, and also some of the ways in which you can imagine, what might break beta oxidation if this enzyme is missing, or chemically, looking at a fatty acid and saying, oh, okay, this can go through this many rounds of beta oxidation, and then it needs to have this done and that done so that it can continue. So those are just some tips when you're going through and you're studying for the exam. Thanks for watching. Stay safe.